Alright, hey everybody. This review that I'm doing now is basically the test review that I did before the test. So hopefully, since it's recorded now, you can kind of watch it as you want. It's going to be pretty long. We're going through about 2.5 pages of material. So I'll make little links at the bottom of the video that'll kind of explain where we're at. And so you can just hop straight to a subject that you want to get to. So the first section is chemistry of life, biomolecules. So we're going to talk about bonds. We're going to identify types, numbers, structure, chemistry, and we're also going to talk about line structure. So I'll do a few examples of those. So we'll just get straight into it. There's two types of bonds. There's covalent bonds, and there's non-covalent. Under covalent, we're going to have polar and nonpolar. So this is an example of a polar covalent bond, which would be those right there. And then something with nonpolar would be these ones right there. All those lines would be nonpolar covalent. Over here we have ionic bonds, we have hydrogen bonds, and then we have van der Waals. So ionic bonds are going to be your things like NaCl, and it's going to be between things that have opposite charges, and when they conjoin, they're going to neutralize the charges. You have H bonds, which they'll look like that. And then you have van der Waals, which I'm just going to draw fatty acid tails. And van der Waal interactions are the interactions between here. And those are due to partial positive negative charges. Remember that, let's say we have an atom here, and the electron, it's just kind of going all over the place. It could be here, it's just going to be anywhere in this general area. Well, when that electron is over here, we're going to have a very tiny partial positive, partial negative. It's going to be extremely small, but just by random chance, these can line up and they can interact with each other. So these tiny partial negatives and positives could line up when you have hundreds of molecules all bumping against each other. Okay, now if you noticed how I drew these, I drew them in order of strength. So a covalent bond is stronger than a non-covalent bond, and so these are kind of about equal. And then you get down here, ionic bond is stronger than a hydrogen bond, and that is stronger than a van der Waals. So understand this, and where you might see questions like this is like the one with the protein. And you might be asked a question, you might have this protein that has all these different, might have some ionic bonds, maybe a couple covalent nonpolar bonds, and you'll be asked which one would require the highest temperature, or the highest level of acid in order to break. And the answer to that would be, in that case, a nonpolar one. So just know your bond strength and how that could affect protein structures. I think it'd be pretty solid. So we'll go and do some line structures. So I'm going to do a couple examples. And I've said it a few times, but the only way you're going to get good at these is just doing a whole lot of them. And if you get good now, then organic chemistry and stuff will be a little bit easier. So here we have 3 examples. Line drawing. Okay, so in the line drawing, every single time this line comes to a point, there's a carbon there. So this one could actually look like this. And so what do we see in this drawing that is no longer there? Okay, well, each of these carbons, so 1, 2, 3, 4, are actually right here. 1, 2, 3, 4. And each of these hydrogens are actually right here. They're just not drawn in. And the reason this is even a thing in science, it's kind of like the language of molecules. And it's literally like learning a language, especially when you get into your higher level chemistry classes. You're going to study it just as if you're studying a language. There's special nomenclature and everything. It just makes it easier. Imagine drawing out this structure every single time. And this is a small structure. This is a butane. So that's a small structure. Most structures in organic chemistry or just in general are way bigger than that. So if you had to draw all these carbons and hydrogens, it would just take forever. So scientists have created this method where all carbons and hydrogens are inferred. So if you see something like this over here, right off the bat, you know there's 1, 2, 3 carbons. So the structure looks like this. And you know that every carbon needs to be attached to 4 different things. And that's because carbon has 4 spots in its valence shell that can make it go to 8 total, which satisfies the octet rule. So C needs 4, N needs 3, oxygen needs 2, which is why oxygen always binds to 2 things, nitrogen binds to 3, carbon will bind to 4. So the best way to solve this is to just count your bonds. So we're going to start with this carbon right here. We see it has 1, 2 bonds. We know it needs to have 4, so that means there's 2 hydrogens coming off. Same with this one. Same with this one. So every time, just go through, count your carbons. Know that every carbon needs to be attached to 4 things. And then if it's attached to 3, there's 1 hydrogen. If it's attached to 2, then there's 2 hydrogens, so on and so forth. So this next one, it's a little bit trickier. We have carbon here, and a carbon here. Some people are a little confused if there's a carbon there or not, and the answer to that is no, there's not a carbon. Anytime this line connects straight to an oxygen, a nitrogen, a bromine, anything, it's just that's what's there. So let's actually draw the structure so you can kind of see it. So this is basically what this looks like. And you're going to have some lone pairs up here on this oxygen. So once again, you just go through. We'll start with this carbon right here. It's hooked to 1 thing, so that means it has 3 hydrogens to bring the total to 4. This carbon right here is hooked to 2, 1, 2, so that means it must have 2 hydrogens. This carbon is hooked to 1, 2 things, so it must have 2 hydrogens. This oxygen, we said oxygen needs 2, is hooked to 1, 2, so it's satisfied. We don't need to add anything to that. And just so you know, this is polar because this alcohol group, so it'd be hydrophilic, whereas this one is all nonpolar, so it's hydrophobic. Okay, so that was bonds. Now we'll go over, we're still in the chemistry of life biomolecule section, so we'll bump down to number 2, and we're going to talk about electronegativity, polar, and nonpolar. So first I'm going to explain what electronegativity even is, and then we'll explain polarity and how that deals with hydrophobicity. So we're going to draw something that's very electronegative. Oxygen and hydrogen. So this oxygen is so electronegative that it's going to hold electron density up towards itself. So these shared electrons are going to be way up here by this oxygen. And so where this hydrogen, its electron cloud used to be like this, now its electron cloud is like that when it's joined with this oxygen. So you can kind of think about how this would affect it if all of a sudden the electron cloud is just barely wrapping around and there's an electron density way up here. This is electron density. Well, the protons of this nucleus are a lot closer to this outer shell. They're a lot closer now. So you're going to get a partial positive. And with this electron density up here, these electrons are a lot further. So those protons aren't going to neutralize the electrons as much. So you're going to get a partial negative. And that's the explanation on why you get these partial charges. Another way to think of this is electronegativity increases this way. Atomic radius increases this way. So as a molecule gets bigger, okay, so we'll say this is Na and we'll say this is oxygen. This distance, so as the molecule gets bigger, this distance is decreased, which means the protons are further away from the electrons. So these protons are very close to the surface, which is why any electrons are going to be drawn to it. There's a lot more proton interaction with electrons. And so that basically explains that. So now we'll talk about polarity and nonpolar. So right off the bat, no, polar equals hydrophilic. Nonpolar equals hydrophobic. So anything that has polarity has, think of poles like the North Pole and South Pole. So if we look here, there's some polarity. We have partial charges. Think of partial charges, think of a large difference in electronegativity. Anything that's nonpolar, we'll draw some examples here. So boom, that's polar. That's polar. So for nonpolar, we have things like this. That would be nonpolar. And remember, if you want to do some practice, count how many carbons and hydrogens are in this. 1, 2, 3, 4. And then we have 3, 6, 7, 8, 9, 10. 4 carbons, 10 hydrogens. So that's hydrophobic. And just know the molecules where you'll find these in. So a lot of our carbohydrates, think of glucose, lactose, all these oses, they're going to have a lot of OHs and polarity. Think of DNA. It has that phosphate backbone. It has the nitrogen sugar. A lot of the base pairs even have a nitrogen in them. But usually the base pairs are actually more hydrophobic in character because the 2 rings that it's composed of, or the single ring. As far as nonpolar, you're going to see like the fatty acid tails on lipids, large carbon structures, some of the amino acids. So just know how that'll affect everything. Know the hydrophobic effect and how that leads to an increase in entropy, which is why it's spontaneous, and why the hydrophobic molecules will find their way to the interior of a protein if they're in a polar environment. Alright, so now we're in macromolecules. So I'm basically just going to draw the table that you guys have been given. It's a pretty simple table. It just has the macro, the mono, and a couple other things. So we'll start with protein. We have carbs, lipids, okay, now some people on the test asked me, because there's an option that said, which of the following, what was the question? Which of the following macromolecules is composed of monomers of nucleotides or something like that? Some people were confused about DNA, RNA, and you knew the macromolecules nucleic acid, but you thought DNA, RNA was the wrong choice. But RNA stands for ribose nucleic acid. DNA stands for deoxyribose nucleic acid. So just a heads up on what those mean. So for the protein, what makes up a protein is amino acids. And when it comes to amino acids, there's hydrophobic, hydrophilic, and then there's charged, positive and negative. So the positives are going to be your acids, and the negatives are going to be your, the positives are going to be your bases, negatives are going to be your acids. Carbs, also known as polysaccharides, are composed of monosaccharides. Your lipids are composed of fatty acids and glycerol. And your nucleic acid are composed of nucleotides. So now we'll talk about the function of proteins. You might see these things in pumps, channels, transporters. For monosaccharides, carbs, the main thing is going to be energy storage and structure. Lipids, it's basically all structure and some energy storage. Think of a triacylglycerol. That's actually one of the main forms of energy storage. So nucleic acids, their function is information storage. And it can store a lot of information. So proteins, it depends what structure it's in. So remember, proteins have primary, secondary, tertiary, and quaternary structure. The primary structure is just the polypeptide chain, and it's basically going to look like it'll basically look like this with R groups coming off of here. You also have secondary, which is your alpha helices and beta plated sheets. And then you have your tertiary, which is like the functioning actual protein. And then you have quaternary, which is multiple tertiaries all hooked together. So let's go to carbs. With your carbs, it could look like this. I remember there's going to be OHs all over these things. The main way to recognize these is by the OHs. You could also see it in something that looks like this. And this is called a chair formation. So it may look something like this. And you also might see it something that looks like this. With your lipids, you're going to see something like this. It could be anything. It could be a phosphorus here, nitrogen, but just know this is what you're looking for. If you see these long fatty acid tails, that's how you know you're working with a lipid. And then nucleic acids, you have your phosphorus hooked to your sugar hooked to your base pair. These three things make the nucleotide. This is a nucleotide. These form together to form nucleic acids, which are the whole double helix long strand. So, sorry, double-stranded helix. Alright, so we just learned about the macromolecules, the monomers, the function, what they look like, and that concludes chemistry of life and biomolecules. So we'll go back through and kind of just talk about what we went over. We went over covalent, non-covalent, and the strength of all these bonds and how that would affect melting temperatures or dissociating a protein. We went over some line drawings. We talked about electronegativity and why electronegativity even occurs and also how it affects things. And we talked about polar, nonpolar, some molecules, how to recognize it, and if that makes it hydrophilic or hydrophobic. Then we went over macromolecules, the monomers that make them, their function, and what they look like. So now we're going to move on to the second section, protein structure and function. So we'll go a little bit more in depth on proteins. First, we're going to talk about the primary, secondary, tertiary, and quaternary structure and the bonds that hold them together. Okay. So your primary structure is just a polypeptide sequence, and it's held together by covalent bonds called peptide bonds. I'm going to switch the color. So what this will look like is basically what we drew earlier. So that RNH3, you'll notice these repeating. You'll basically notice this repeating group. And you'll have all these R groups sticking out. And remember, the R groups are what controls the chemistry of a primary sequence. If there's a lot of hydrophobic, as it's coming out of the ribosome is what actually builds these. The ribosome is, the primary sequence is going to start curling up and folding, and that's all due to the hydrophobic effect. For this secondary, we have alpha helices and beta plated sheets. And these are made of hydrogen bonds between carboxyl and amide groups. So just know that these are between this and this, and not the R groups. Now for tertiary, every single bond is basically activated. We have ionic, hydrogen bonds, van der Waals, and then we also have covalent bonds such as sulfide bridges. And these sulfide bridges can be disrupted. You can disrupt these covalent bonds without disrupting these covalent bonds because it's not as strong. And you guys actually do this every day, most of you probably. When you take a shower or you wash your hands, look at your soap, especially a lot of shampoo has sodium lauryl sulfate in it, which is a detergent. I'm sure even your washing detergent has it. And it helps break up those bonds of proteins and kind of get them off your body. So quaternary is going to be everything that tertiary is. It's just multiple tertiary structures all bound together. So next we're going to talk about amino acids and where you would find them. So we have this polar solution and this non-polar solution. And these are our proteins. These are our proteins. So the best way to memorize this is that the exterior, so this is all polar, and this is all non-polar. The exterior is going to want to match whatever solution it's in. So all these exterior amino acids are going to be your polar ones, and then inside you're going to find all your non-polar, aka hydrophobic ones. Over here, this whole inside is going to want to match this. So you're going to find all your non-polar, and inside you will find your polar amino acids. So just know when you throw, let's say you get some random protein, or a protein is pumping out of a ribosome, and if it's pumping into a polar solution, the polar amino acids will face the outside of the complex. The non-polar will face the inside, vice versa. So that's the hydrophobic effect. So next, for number 5, it says predict how changing amino acids impacts protein structure or function. So think of if you had this protein that it really needs its negatively charged acidic amino acid residues in order to perform some function that's very important to life. Well, if all of a sudden there's some sort of coding sequence or some sort of error mistake during the transcription or translation, it could result in the protein getting, let's say, positive base nucleotides instead, or amino acids instead. So now we just went from a negative to a positive. It's still polar, but it might have an opposite charge, so maybe it's going to repel from the active site where the other one attracted. So just be able to conceptualize in your head how changing an amino acid can change function. And if you're asked this question, look at what the initial amino acid is. Is it polar? Is it non-polar? Is it positive, minus? And then look at what we're switching it to if you're asked that in a question. And just think about it and try to conceptualize, like, okay, is that going to make it repel? If your initial amino acid was positive and it's interacting with an active site, well, the active site is probably negative. So now if we switch the amino acid to negative, there's going to be repulsion. If we switch it to hydrophobic, it's probably not even going to find that little nook inside the active site. So be able to conceptualize stuff in your head. It's going to help you out a lot. Okay, so next we're on 3, so we're actually going through this pretty fast. So we're on cell membranes and transport. We'll talk about the cell membrane and selective permeability. Alright, so you have the cell membrane. We're just going to draw it like this. Alright, so this is our cell membrane. Now the big thing to understand here is your regions. So we have these very small regions of polarity, and then this huge region of hydrophobic. So polar molecules are probably not going to make it through this hydrophobic region too easily. So right off the bat, you know that hydrophobic molecules can get through this easier. So what's going to go all the way through? Small nonpolar, they're going to go right through. They're going to easily pass through. What's going to have a kind of okay chance passing through is going to be small partial charges. Think of H2O, where this would be like O2 into H2, all your gases. A large nonpolar would be something like a steroid. And then what's just going to not even have a chance at making it through, that's going to be charged atoms like Na plus or Cl minus, and your large polar like glucose. They're going to need things like a transporter or some sort of pump. While we're talking about transporters, let's talk about why something would even need a transporter. So we're going to draw the same thing. This is my lipid membrane. This is my pump. It's not a pump, it's a channel. And we'll say we want to get this through here. Okay, so why do we even need a channel for this positively charged ion? Well, we know this entire, let me draw this. This entire inside is all hydrophobic. So this potassium isn't just going to pass through. We need this channel. And what this channel is going to do is it's going to make these walls that face the inside of the membrane hydrophobic. But the inside, this whole inside right here, is all going to be hydrophilic. And now that it's hydrophilic, this can pass through. So just know that's how a channel builds this defensive barrier against the hydrophobicity of the inside of a cellular membrane. So next we'll talk about active and passive transport. So active is low to high. Passive is high to low. Yeah, this can also be diffusion. So the two types of diffusion, you have passive and you have facilitated. Passive, you're going to notice with things like gas, hydrogen, small, hydrophobic. And that's because they can just pass right through the membrane. So they're always going to go from high to low concentration, and they can just slip right through the membrane. For facilitated, it'll be for things like large, hydrophobic possibly. But more importantly, it's going to be for your polar, things like glucose, maybe water, things like that. Now active, there's primary and secondary. Primary is going to be things like pumps, they use energy directly. And an example is sodium potassium pump. It uses 1 ATP to pump the sodium and potassium. Secondary, you have symporter and antiporter. So what both of these do is they use the energy from an existing concentration gradient that happens spontaneously, and there's a little bit of excess energy that happens with that spontaneous reaction, and it creates enough energy to bring one thing with it. So we'll think of what is spontaneous? This high to low is spontaneous, and it's going to create energy. This low to high is not spontaneous. And it's going to use the energy created from this high to low to bring this one in. The reverse of this is antiporter. So and to clear up any confusion, we'll draw a picture. Boom. So this is a picture of this going from high to low, and it's going to create enough energy to bring this one from low to high. If you could flip this, and we'll erase all this. If you were to do this, this is now an antiporter. So we just covered active and passive transport. All right, and I think that should about do it with this. We'll go back through and kind of talk about what we just mentioned. So we just covered a lot of big concepts. Go through this, take your time, understand it all. Get in the Cogbooks. Cogbooks is an amazing, I wish I had that when I took this class. It really has a ton of good information in there, and it's really streamlined. It's a lot quicker than reading a textbook page by page. So we went over covalent, non-covalent, all the bonds that reside under those two umbrella terms, and the strengths of those bonds. We went over line drawing, polar, non-polar, how to recognize it. Then we went over all the macromolecules. We went over protein structure, what holds them together, and how to recognize them. Then we went over proteins and how they spontaneously fold in a solution, and how you can know what's going to fold where. Is it going to be hydrophobic amino acids on the outside or hydrophilic? We then went over lipids and these membranes and what can pass through them and what can't. We also mentioned why these can't pass through. And then we mentioned something called a channel, which can enable them to pass through. Then we went on to active and passive transport, and we talked about passive, which is simple diffusion, and we talked about facilitated diffusion. We then went on to active transport, where we talked about primary and secondary, what the difference is, and we talked about symporter, antiporters, and how those actually they take advantage of an existing concentration gradient to move another molecule against its concentration gradient. So if you guys need any more help on anything, just send me an email or whatever. It's pretty easy to make a video on this, so just let me know.